Okay, so far we read about how the receptor mediated endocytosis work, how does a coated pit is formed, what the coated pit is coated with, how does a vesicle created, how does the vesicle travel towards the the endosomal compartment, how it is acted upon by rab protein, etc. So let's see now what happens at this compartment where we have our hormone, that is, or a ligand, that is brought by this vesicle. So phagosomes and pinocytotic vesicles typically fuse with lysosome within the endosomal compartment for digestion of their content, while molecules enter by receptor mediated endocytosis may be directed down other ways pathways. The membrane of many late endosomes have ATP driven hydrogen pump that acidifies their interior, activating the hydrolytic enzymes of lysosomes, and in other endosomes, some endosomes causing ligands to uncouple their receptors, after which the two molecules are sorted into separate endosomes. Okay. The receptor may be sorted into recycling endosomes and returning to their cell surface for reuse. Low density lipoprotein receptors, for example, are recycled several times within cells. Other endosomes may release their entire content at a separate domain of the cell membrane, transcytosis, which occur in many epithelial cells. So, firstly, there is the protein that has been coupled with the receptor protein that has been coupled with the hormone. What happens is they are decoupled, and this receptor protein goes back to the cell membrane for creating a new pit, a new uh, it's ready for the new invagination and for creating new vesicle. Now that this hormone or a ligand that is here is sometimes involves and activates lysosome and undergoes a reaction with lysosome. We'll study later in lysosome how it reacts and what how dynamic this lysosome is. But the other way is that the uh, content of these vesicles travel to the other part of the cell, and they just diffuse it out of the cell. This type of process, where a vesicle goes from one end to the other end and exocytose, is called transcytosis. So movement of I think here the most important part was the membrane of late endosomes have ATP driven hydrogen pump that acidifies their interior, activating the hydrolytic enzymes of lysosome. We will study what hydrolytic enzymes do in lysosomes and how it affects the selectivity. We'll study lysosome as an organelle in the later part. We also need to know that these are mostly low density lipoprotein receptors. That are usually recycled several times. So this can just be a question. So we can have this highlighted that low LDLs are the one that can be recycled several times within the cell. Okay, so movement of large molecule from inside to outside of the cell usually involves vesicular transport in the process called exocytosis, as I already mentioned about exocytosis. In exocytosis, a cytoplasmic vesicle containing the molecule to be secreted fuse with the plasma membrane, resulting in the release of its content into the extracellular space without compromising the integrity of the plasma membrane. So, in exocytosis, a cytoplasmic vesicle containing the molecule to be secreted fuse with plasma membrane. We can just highlight and we'll know what exocytosis is. So basically, what exocytosis means is a vesicle goes up to the plasma membrane, and the coat of this vesicle gets fused with the plasma membrane, and the material inside the vesicle is removed out of the cell. Exocytosis. Exocytosis triggered in many cells by transient increase in cytosolic calcium ions. We need to know which ion increase causes this cytosolic effect causing exocytosis. Membrane fusion during exocytosis is highly regulated with selective interactions between several specific membrane protein. We need to know that it is highly regulated procedure. Okay, exocytosis. So exocytosis of macromolecules made by cells occur via either two ways. There are two ways: consecutive secretion and regulated secretion. Consecutive secretion is used for products that are released from cell continuously as soon as synthesis is complete, such as collagen subunits for the extracellular matrix. So we will just highlight that all the products that are released continuously are exocytosed by a pathway called consecutive secretion, because like collagen, as an example, collagen makes a big component of extracellular matrix. And we will study, by the way, the contents of extracellular matrix, the different collagen fibers in the later chapters. So but for now we need to know that collagen is a subunit of extracellular matrix and is exocytosized by consecutive secretion. We have regulated secretion. Regulatory secretion occurs in response to signal coming to the cell, such as the release of digestive enzymes from the pancreatic cells in response to the specific stimuli. Regulated exocytosis of stored products from epithelial cells usually occurs specifically at the apical domain of the cell, constituting a major mechanism of glandular secretion. See chapter 4. We will study again this in chapter 4 about the signal regulated exocytosis. For now, we just can highlight that regulated secretion occurs in response to signals coming from the cells, such as release of digestive enzymes, pancreatic cells, etc., with a specific stimulus. So, this type of exocytosis is often found in all the hormonal glands, etc. So, we need a stimulus, and when we have a stimulus, only then is exocytosis takes place. We will study chapter 4 and we will know that what types of cells are the ones that uh, carry out regulated exocytosis, regulated secretion. Uh, Portion of the cell membrane becomes part of the endocytosis vesicle or vacuoles during exocytosis. During exocytosis, so far we know that plasma membrane is the most important part when it's come to transport, because exocytosis beat endocytosis, we need the plasma membrane to participate in the completion of this procedure. So like endocytosis, also for the exocytosis we need plasma membrane, the cell membrane, to participate. During exocytosis, membrane is returned to the cell surface. This process of membrane movement and recycling is called membrane trafficking. So this involvement of the plasma membrane with exocytosis and endocytosis is called membrane trafficking. We can highlight this whole part, what membrane trafficking is. Basically, portions of the cell membrane becoming part of the endocytotic vesicles. Like in the coated pit, we created a vesicle that was actually a part of plasma membrane, and same happens with the exocytosis, where this part of the membrane goes and adjusts back to the cell membrane, and this all process is called membrane trafficking. Trafficking of membrane components occur continuously in most cells and is not only crucial for maintaining the cell but also for physiologically important processes such as reducing blood lipid levels. So what this is the only important part, that it reduces blood lipid levels. What? Membrane trafficking. In many cells, subpopulations of vacuoles and tubules within the endosomal compartment accumulate small vesicles within their lumens by further invaginations of their limiting membranes, becoming multivesicular bodies. While multivesicular bodies may merge with lysosomes of selective degradation of their content, this organelle may also fuse with the plasma membrane and release the intraluminal vesicle outside the cell. The small vesicle released are called exosomes. So, like in many cells, subpopulation of vacuoles and tubules within the endosomal compartment accumulate small vesicles within their lumen by further invagination of their limiting membranes, becoming multivesicular lobules. We need to focus a bit here about what are multivesicular bodies. While multivesicular bodies may merge with liposomes for selective degradation of their content, so now we know that there is a subpopulation of vacuoles and tubules that were released at the endosomal compartment. Now, these small vesicles within their lumen, by further invagination, so a vesicle undergoes an invagination again, becomes a multivesicular body. While multivesicular bodies may merge with lysosomes for selective degradation of their content, this organelle may also fuse with the plasma membrane and release the intraluminal vesicle outside of the cell. So now these small multivesicular bodies have their own contents, the ligands, the hormones, anything. If required, they will go and give this content merging with the lysosome. If not required, it will go again back to the cell membrane and release this content outside of the cell. The small vesicles that are less than, let's say, 120 nanometer diameters, they are called exosomes. These multivesicular bodies basically are called exosomes, which can fuse with other cell, transferring their contents and membranes. So they can go to fuse with lysosomes, or they can go and just fuse with membrane, the cell membrane. Let's now see signal reception and transduction. So we saw that how are the receptors receive this signal from a ligand, from a hormone, and we will just read how this signal reception and transduction takes place. Cells in the multicellular organism communicate with one another to regulate tissue and organ development, to control their growth and division, and to coordinate their functions. Many adjacent cells form communicating gap junctions that couple the cells and other exchange ions and small molecules. So we have a gap junction now, which is so important. We will study all the junctions. I mentioned this before as well, that we will study all the junctions in the later chapter, chapter 4. But we need to understand that for transport and communication, junctions are very important. We have a lot of junctions, tight junctions, gap junctions, etc. But the most important role of coordination is carried out by gap junctions. Cells also use about 25 families of receptors to detect and respond to various extracellular molecules and physical stimuli. I told you already about these receptors. They are the proteins on the plasma membrane. They are only integral proteins. Cannot be peripheral proteins. So these receptors, they detect and respond to various extracellular molecules and physical stimuli. Each cell type in the body contains a distinctive set of cell surfaces and cytoplasmic receptor proteins that enables it to respond to a complementary set of signaling molecules in a specific programmed way. So these receptors are specialized. There are 25 families altogether of these receptors, but each cell has its own types of receptors that is required for its particular function, and they are very selective and very specific, and they react to very specific ligand, and they work in a programmed way. So cells bearing receptors for a specific ligand are referred to as target cells of that molecule. So These cells, which bear a specific ligand for a specific receptor for a specific ligand, they are called the target cells for that specific ligand or that specific molecule. The route of signal molecules from source to target provide one way to categorize the signaling process. Let's see about how the signaling process works. In endocrine signaling, the signal molecule here called hormones, because endocrine, so we will talk about hormones here, are carried in the blood from their source to target cells throughout the body. We know how hormones travel within the blood. So in endocrine signaling, the signal molecules here are called hormones, are carried in the blood from their sources to target cells throughout the body. In paracrine signaling, endocrine we know. Now the paracrine, the chemical ligands diffuse in the extracellular fluid but is rapidly metabolized so that its effect is only local on target cells near its source. Paracrine, these are types of also secretions. Paracrine secretion is type of secretion where it is just secreted, it secretes its own material in its own environment. So in that case, we not need to travel these secretions. In the same way, in paracrine signaling, where we have this chemical ligands that diffuse in the extracellular fluid around the area where they are already secreted, and they just create a response and they are specific to the targeted cells in that area. This type of signaling is called paracrine signaling. In synaptic signaling, a specific kind of paracrine interaction neurotransmitter acts as a adjacent cell through special contact areas called synapses. So there is a neurotransmitter, a special kind of paracrine interaction that takes place here, uh, and this neurotransmitter goes and acts on the other cell. We will read about synapses in chapter 9 much in detail, and how the neurotransmitters secreted from because these neurotransmitters are also secreted in the form of vesicles. So we need to and we need to understand, as we are reading about the vesicle transport, so we need to know that. But neurotransmitter secretions and their movement is explained much in detail in chapter 9 about nervous tissue. So we will really read about synapses and movement of neurotransmitter there. But this type of movement, this type of transduction of the neurotransmitter is called synaptic signaling. Okay, we decided that we will read table 2.2, where we have mechanism of transport across the plasma membrane, and everything with an additional information has been all clubbed in this table. We will just go through it and try to remember in our words that what do we remember from what we read today. So firstly, two types of transport: the passive one, the active one. Passive one does not require energy, active one requires energy. Also, passive one is always towards the concentration gradient, whereas active process can also go against the concentration gradient. Now there are different types of passive processes. Diffusion, majorly we read about three: diffusions, channel mediated, and carriers. So diffusion, channels, and carrier. Diffusion is basically movement from higher concentration to lower concentration, but not in the case of water. Water moves slowly. We also saw the channel mediated. Channel mediated is also through the concentration gradient, but these channels are specific for specific ions. Three of them we read mostly. We have as an example Na plus here, we but read Na plus, Ca2 plus, and we need to go back and check. I remember reading about Ca2 plus, Na2 plus, uh, Na plus and Ca2 plus. Right, sorry, my bad. Na plus, Ca2 plus. So, and next is the carrier. Carrier is basically the movement of protein that is like a flipping movement, and it's also down the concentration gradient by a carrier protein. We also need to remember the carrier protein for uh, the porins, aquaporins, that were responsible for movement of water with this specialized protein, aquaporins. We also know osmosis. Osmosis is basically diffusion of water. So water movement from higher concentration to lower concentration is called osmosis. Now let's check about the active processes. These were the only passive processes. Active processes, we read about primary, secondary, symport, antiport. This is an additional information. We already have read about how the active transport works, but let's check out what are the different types of active transports. So primary is movement of substance up its concentration gradient directly with the help of adenosine triphosphate. But in the secondary transport, movement of substances up its concentration gradient is powered by harnessing the movement of second substance down its concentration gradient. So basically, to move a, a let's say a substance from lower concentration to higher concentration, we need to bring something from higher concentration to lower concentration, that and it has to be some other second substance, and we have to bring it down its concentration gradient so that we have power, the energy to move something from against the concentration gradient. Another method of active transport is called symport. Symport and antiport, very similar things. Symport is movement of substance up its concentration gradient in the same direction as Na plus. Whenever a sodium moves, we have to remember about sodium. Whenever we are talking about symport and antiport, we always remember that we are talking about sodium ions mostly. So symport is the movement of concentration gradient, but it but it has to be in the same direction as sodium. They both will move together. And in the antiport, it has to be opposite to the sodium channels, the sodium direction. We have an example, very very important. Whenever a sodium moves, let's say from out of the cell to inside of the cell, we can move glucose inside of the cell to outside of the cell, even if we have a higher concentration outside. Doesn't matter, we can go against the gradient, because this is an active transport, we can go against. And in the case of antiport is an example of Na plus and H plus. So whenever sodium comes in, glucose also comes in. They both needs to be in the same direction when we are talking about symport. Antiport is an example where Na plus goes in and H plus comes out. Simple. Vesicular transport, we talked the most about vesicular transport. We talked about exocytosis, endocytosis, phagos, and the types of endocytosis are three: uh, phagocytosis, pinocytosis, and receptor mediated endocytosis. We talked the most about these much in detail, and we should not touch this anymore and move to the next topic. So let's go and touch some new topics. So we read about signaling, and we read about the endocrine signaling. Here we read about the paracrine signaling, we read about the synaptic signaling. The next type of signaling is autocrine signaling and juxtacrine signaling. Autocrine signaling signals bind receptors on the same cells that produce the messenger molecule. So it's basically a secretion, but for the same cell. A cell secretes a vesicle that is required by himself, and this type of secretion is called autocrine secretion. In juxtacrine signaling, it's important in early embryonic tissue interactions. The signaling molecules are cell membrane bound proteins which bind surface receptors of the target cell when the two cells make direct physical contact. So for this two cells, the target cell and the producing cell, needs to be in physical contact. In that case, we can just cell membrane bound proteins can be transferred to the target cell by making a direct physical contact. This is important for juxtacrine signaling. Okay, so receptors for hydrophilic signaling molecule, including polypeptide hormones and neurotransmitters, are usually transmembrane proteins in the plasma lemma of target cells. Three important functional classes of such receptors are shown in the figure 2.8. So, receptors for hydrophilic signaling. Hydrophilic means, the word hydrophilic means water loving. Hydrophobic means water repelling. So, hydrophilic signaling molecules include polypeptide hormones, means protein hormones, and neurotransmitters. So, let's see the three important functional class of this type of receptors. So, the target cell has three different types of receptors that will react with the protein hormones and the neurotransmitter. They are channel linked receptors, enzymatic receptors, and G protein coupled receptors. So channel linked receptors. So now we learned about how a vesicle is moved out, exocytosis, and this signaling molecule goes to the target cell. Now let's see what are the types of receptors that are present on that target cell. And there are three types of receptors, as I said. So first one, channel link receptors, associate open associated channels upon ligand binding to promote transfer of molecules or ions across the membrane. Simple channels, we know a, a protein molecule or neurotransmitter comes, binds to the receptor protein, and is moved through the channel into the cell, like the same pathway for ions. Enzymatic receptors, in which ligand binding induces catalytic activity in associated peripheral proteins. So in enzymatic receptors, this ligand bind induces a catalytic activity, and that is associated with peripheral protein. So these enzymatic receptors are basically integral proteins, but they're associated with the peripheral proteins. They are not peripheral proteins, and we need to highlight about this catalytic activity. So it induces the catalytic ligand binding in the enzymatic receptor. Basically, what do enzymes do? Most important role is enzymes plays as a catalyst for most of the reactions in our body. So they have to induce the catalytic activity. G protein coupled receptors upon ligand binding stimulate associated G protein, and G protein, which then binds to GTP, are released to activate other cytoplasmic proteins. So GTP, guanosine triphosphate, the nucleotide, will go and activate other proteins. Like, we know the basics of DNA to RNA, RNA to protein. How can GTP activate a polypeptide? And but we need G protein to be activated, and they are activated by G protein coupled receptors. They stimulate associated G protein, activate other cytoplasmic proteins. Perfect. Perfect. So we read about three types of receptors that receive a particular signal. Ligand binding such receptors in cell membrane can be considered first messenger, beginning a process of signal transduction by activating a series of intermediary enzyme downstream to produce changes in the cytoplasm, the nucleus, or both. So this type of ligand now will be responsible for the signal. This signal, what does this signal do? They stimulate an enzymatic activity, resulting in what? Resulting in cytoplasmic changes or changes in the nucleus, or both together. So this is how a signal is produced, how it's transferred, how it's received at the receptor, and how does the changes takes place in a cell. Channel mediated ions influx or activation of kinases can activate various cytoplasmic proteins, amplifying the signal. Activated G protein target ion channels or other membrane bound effectors that also propagate the signal further into the cell. One such effector protein in the enzyme is the enzyme adenyl silase, which generates large quantities of second messenger molecules such as cyclic adenosine monophosphates. So the first signal, we know that it traveled through in the form of ligand, but we later, when it, other than going and producing changes in the cytoplasm, the activated G protein target ion channels, they can further create a one more signal that will be used for some more specific works and can be called a secondary messenger molecule, that is the CAMP, that is cyclic adenosine monophosphate. What is it? It's a secondary messenger molecule, and it is it is basically it is basically you know activated by this protein that was activated by the G protein. So G protein binds to the GTP that releases something. It's a GTP releases and G protein releases. I'm sorry. G protein releases GTP. This GTP in turn activates a protein. Now, what this protein does is that it activates the enzymatic activity of adenyl cyclase, converting it into CAMP, cyclic adenosine cyclic monophosphate, cyclic adenosine monophosphate. This is a second messenger. Let's see what other second messengers we have. Other second messengers include 1,2-diacetyl glycerol and IP3, that is inositol 1,4,5 triphosphate. We only important part here is to remember what are the three main important secondary molecules, the second messenger molecules. First messenger is always the ligand that came to the cell, created the first signal, but created the signal, and the protein that was at the end stimulated creates a second signal again through three molecules, three types of proteins. Which are the three? DAG, IP3, we will just highlight these all. DAG, IP3, and CAMP, cyclic adenosine monophosphate. Okay, never mind. Okay, the ionic changes or the second messenger amplify the first signal and trigger a cascade of enzymatic activity, usually including kinases, leading to changes in gene expression or cell behavior. So basically, why is a second signal required? Just to have a larger impact, just to have a more larger impact. Mostly, it usually increases the kinase work, and basically it helps in leading to changes in cell expression and cell behavior. That is what we wanted. The signal came to cell for only one reason, to change a particular behavior. Second messengers may diffuse through the cytoplasm or be retained locally by scaffold proteins for more focused amplification of activity. Scaffold proteins. From the previous lecture, we know what are scaffold proteins. Comment down if you can pause and comment down what are scaffold proteins here. So scaffold proteins are basically the proteins that we saw in the rafts, the lipid rafts that we saw on the plasma membrane. You can go back and check what the lipid rafts and how the enzyme complex that we talked about works and what are these lipid rafts. But the protein that make this complex are the scaffold protein on the plasma membrane. Low molecular weight hydrophobic signaling. Let's also highlight this, that these secondary messengers can also move out of the cell or can be retained inside of the cell locally by the same scaffold protein. Low molecular weight hydrophobic signaling molecule such as steroids and thyroid hormone. this is important. Signaling molecules are not just hormones always or ligands, but we have a low molecular weight hydrophobic signals. We know hydrophilic signals too, neurotransmitters and protein hormones. Now we have hydrophobic signals. Hydrophobic is water repelling signals. These are steroids and thyroid hormone. Very important. We should go and highlight the part where we saw what are hydrophilic signaling molecules, because this can be compared and asked to you. Okay, what do they do now? Bind reversibly to carrier protein in the plasma membrane for transport through the body. Such hormones are lipophilic. Philic is loving, lipo is lipid, and pass by diffusion through cell membrane. Lipophilic, whenever it's we have a lipophilic molecule, is easy to pass from the cell membrane for because of its bipolar amphiphytic uh, behavior. So such hormones are lipophilic and pass by diffusion through cell membrane, binding to specific cytoplasmic receptors, protein in target cells. With many steroid hormones, receptor binding activities that receptor enable the complex to move into the molecule and bind with high affinity to specific DNA sequence. So these hormones, these steroid hormones, they activate the receptors, enabling a complex to move into the nucleus. What does this complex moving into the nucleus do? It binds to specific DNA sequence. This generally increases the level of transcription through those genes. Each steroid hormone is recognized by very different member of family homologous receptor protein. Yes, so we know this, that how fast signaling. So basically all goal for the signal is to change the cell behavior. Specifically when we are talking about steroids, they go directly to the nucleus, to the DNA, and in, they just speed up the process of transcription and translation, and these new generated proteins will create the behavioral change. But in the other hand, when we had the messengers that were hydrophilic, they go and change the activity of the protein that is already created through the translation and transposition. So it is always bigger in number. We have to, it takes time. It's a so comparatively slow process. So this is, I think, important. With many steroid hormones, we just know that what does the steroid hormones do. They go to the DNA sequence and they speed up the process. Okay, so we now know what cell membrane is. Let's now read what consists inside the cell membrane. It's a whole world. What is the different organelles in the cells, and what are organelles, and how do they work? I think it's good time to just have a stop at this lecture, and so that we can just study all